Dear students, the last chapter we have concluded microbes in human welfare. Now we have to go on to the next chapter, applications of biotechnology. That is chapter 9 in biozoology and chapter 10 in zoology accordingly. Now we already studied, that is what we have the principles and process of biotechnology in botany. And based on this one, I don't want to touch once again the principles and processes, but we have to go for only the applications, the uses, how far the technology has been used. That is today. So normally biotechnology, a new term has been given, what is called, that is industrial science. Industrial science. Now, here we have to talk about with what is genetic engineering. What are the events included under genetic engineering? And then we have to go for biotechnology, the definition, and go coin the word, etc. Now, genetic engineering means manipulation of genes according to our will, according to our desire. Manipulation means either synthesis or repair, either removal or addition. All events together called as manipulation. Synthesis, addition, that is removal, then also what we call the repair of DNA, that is called manipulation. Now here, as per the lesson, we have to go for genetic engineering involves three major processes. Number one, manipulation of DNA, that is synthesis of DNA, repairing of DNA, okay, removal of just unwanted DNA, addition of useful DNA. All these events together call as manipulation of DNA. The second one, naturally occurring processes. The naturally occurring process normally in the cell, nothing but you see that one protein synthesis. So this protein synthesis is normally used actually for a wide range of applications. Mainly the therapeutic proteins may be synthesized. Therapeutic proteins means normally the medicinal value proteins. So here protein synthesis, naturally occurring process in the cells because of transcription, translation we already studied under molecular genetics. Okay, so here we are producing proteins which are of therapeutic value, that is a medicinal value. For example, interferons, it's a glycoprotein. Okay, like that, we will see later. Now the third one, recombinant DNA technology. What is recombinant DNA? So I mentioned already manipulation of DNA. Here, a gene of interest, a gene of known function. A gene of interest is nothing but gene of known function. Say an example of insulin gene. That is called gene of interest or gene of known function. So we are extracting that gene from what we call the beta cells of islets of Langerhans and transferring the gene to another organism. The organism may be of same species or different species. And the resultant product is called recombinant DNA. The resultant product is called recombinant DNA or rDNA. The technology is called recombinant DNA technology. It is coming under what we call genetic engineering. So manipulation of genes involves we have manipulation of DNA, synthesis of actually proteins for therapeutic value, and number three, recombinant DNA technology, the production of rDNA by transferring a gene from one organism to another organism with the help of a vector or without vectors. We have studied, I think so. Okay, the technology is called as recombinant DNA technology. Now what is biotechnology? It can be defined as the applications of scientific and engineering principles to the processing of material by biological agents to provide goods and services. Simply harvesting of beneficial products for the benefits of human beings. That's why I said it's also called industrial science. So it can be defined as the applications of scientific and engineering principles to the processing of material by various biological agents to provide goods and services to whom? That is human beings. So we are getting beneficial products by means of biotechnology. That's why it's called as industrial science. Now this lesson includes the applications of biotechnology. We have studied the applications of biotechnology in various fields. So the applications of biotechnology includes number 1, applications in medicine. Number 2, gene therapy. We will discuss one by one. Number 3, stem cell therapy. Number 4, molecular diagnosis. Number 5, biological products and their uses. Number 6, transgenic animals. Animals having their own genome along with the genome of some other organisms. For example, golden rice. And finally, animal cloning. Production of cloned animals, like for example, Dolly, the sheep. So, etc. These are all the things we are going to study in this lesson. Now, biotechnology has its application in the production of medicine. The recombinant DNA technology led to the production of, that is, various hormones and the proteins which are of medicinal value. That is called therapeutic value. Therapeutic value means medicinal value. Okay, we'll discuss one by one. The first one, applications in medicine. Applications of biotechnology in medicines, under which we have to study the production of human insulin hormone by our DNA technology. Okay. Now let us consider the first application of biotechnology, that is application in medicine. And this one, the first one, recombinant human insulin production. How is it produced? Through DNA technology. Before entering into, that is actually the process. Now, what is the role of insulin and what is its chemical nature? Now, human insulin is synthesized by the beta cells of islets of Langerhans of pancreas. You see that one, the pancreas is a heterocrine gland having islets of Langerhans as hormone secreting endocrine cells. Now, if you are taking the chemical nature, it is formed of 51 amino acids. The 51 amino acids are arranged in the form of what we have two polypeptide chains. One is what is called A chain, another one is called the B chain. Okay, so also calls what is known as a glycyl chain and phenylalanine chain, like that, release both. Now totally, how many amino acids are present in insulin hormone? That is 51. After 51, A chain has 21 amino acids and the B chain is formed of 30 amino acids. The two amino acids are joined together by means of a disulfide bridge or bond. Suppose this is A chain, this is B chain. We have these two are connected by means of what's called disulfide bridge or a bond. The two chains are connected by means of disulfide bond. Wherever you have what is called cysteine residues are present. Now, sulfur containing amino acids only two. In two amino acids only we have sulfur. They are called sulfur containing amino acids. Okay, one methionine, another one cysteine. So these are the two amino acids which contain sulfur containing amino acids. Out of the amino acids found in nature, these are the only two amino acids which have sulfur. So wherever we have cysteine present, suppose for example here the disulfide bridge is formed. Here cysteine molecule is present, here cysteine molecule is present. The bridge is formed only between the cysteine in the case of insulin hormone. And we have studied already the Y-shaped molecule, the immunoglobulin. There also we have what is called actually, that is disulfide bridge. And there is also we have the cysteine residues. The same manner we have the bond is called SS bond or disulfide bond. Now what is the role of insulin? Now the insulin is responsible for another one controlling the sugar. Role of insulin, if you are taking that one, it controls actually the blood sugar level. And in what way? It increases the uptake and utilization of glucose by the cells. So the cells take glucose and then what? They oxidize the glucose to release energy. That's about the role. Controls the blood sugar level. How? Increases the uptake of glucose and the utilization of glucose by the cells. That results in the production of energy. Suppose what would happen if there is any deficiency of insulin hormone? Let's see now. Deficiency of insulin leads to, that is, increase in blood glucose level or increase in glucose concentration in the blood, ultimately results in a disease what's called diabetes mellitus and a complex symptoms. If all the symptoms are not treated, the symptoms too, along with the diabetes mellitus, the results are leading to death also. So, we have actually a continuous program of insulin dependence is required for the treatment of disease. Once a diabetes occurs, it is a chronic disease lasting forever till death. The person has to depend on insulin hormone orally, taking artificial synthesis of insulin to reduce the blood sugar level. That's why I say a continuous program of insulin dependence is required to treat diabetes. You have to take on the tablets to just control the sugar, or you have to take insulin hormone intravenously. Now, in the early days, suppose if there is a deficiency of insulin in the body, to treat diabetes, now the insulin hormone was extracted from, that is, islets of Langerhans of pancreas, either from cow, that is called bovine insulin, or from pig, that is called swine insulin, to treat diabetes. But there is a minor difference between the animal insulin and what is called the human insulin. If you are comparing the two, the animal insulin is slightly different from what is called human insulin. So when it is being given to a patient with the diabetes, sometimes in some cases allergic reactions are resulted. So then there is a need for that one to develop a new technology for the production of insulin hormone. So the insulin hormone is now produced by means of RNA technology. The production of insulin through RNA technology or recombinant DNA technology was started in the late 1970s. Okay, let's continue further. The production of insulin through recombinant DNA technology started in the late 1970s. That is particularly a person, Best and Banding, extracted this actually the hormone from the dog and then just demonstrate their effectiveness. You'll see that one later now. The polypeptide chains I mentioned A, B and C. Normally it is formed of two chains A and B. The two chains are linked together by means of C chain. Now I mentioned about the A chain is formed of 21 amino acids. The B chain is formed of 30 amino acids. Now these two are linked by another chain of 33 amino acids called the C chain. And if you are taking actually the chain, for example in the case of actually the B chain or we have the A chain, we have a preceding sequence. So normally these three chains are produced as a precursor. The precursor is called normally what is called Prepro Insulin. Now this Prepro Insulin is formed of, that is, A and B segments linked by a third C chain preceded by a leader sequence. You see that one? Now the A chain with its amino group is connected to the carboxyl group of the B chain. I show that one. And we have what is called the signal protein, otherwise called as a leader sequence. Then the Prepro Insulin gets converted into proinsulin by the removal of what is called the signal protein or leader sequence. Later what is happening? The leader sequence is removed by means actually after we can say translation. I mentioned here just by, but after translation process. We have transcription and translation what we studied during protein synthesis. So the leader sequence is removed after translation and forms what is called a proinsulin. The first one, pre-proinsulin. After the removal of what is called actually this leader sequence, it is called proinsulin having A and B plus C. Now the C chain is excised, removed to form what is called insulin hormone with A and B chains only. So pre-proinsulin is formed of just we have A, B, C chains plus leader sequence. Then proinsulin is formed of A, B and C only. The leader signal or signal protein and the leader sequence is removed. Then it's called proinsulin. Then by removal of what's called C chain, now A and B chains are linked together by means of disulfide bridge, leading to the formation of insulin. Okay, so this is a process of formation. There is insulin, there is a second product, and before that we have pre-proinsulin. I'll write it down later now, one of space, after removing these statements. Okay, right. Now the leader sequence is removed after translation process, and then the C chain is XII removed. Then we are getting also the leaving A and B polypeptide chains which are linked together by means of disulfide bond, resulting in human insulin hormone. I mentioned already we have what is called the precursor. The precursor is called what is known as Prepro Insulin. After it is being multiplied with the bacterium and being produced in a fermentation tank. Now the next process is extraction and processing of insulin. Now all the three chains we have actually from a precursor. The name of the precursor is called Prepro Insulin. Now the Prepro Insulin consists of the two segments A and B linked by C chain along with a leader sequence. The leading sequence responsible for signaling molecule to just a continuation. So it's also called signal protein. So A chain, B chain is somewhat larger because it's formed on, you see that one, that is 30 amino acids, this is 21, this is formed of 33 amino acids. Okay, now the Prepro Insulin has 4 components. Now after the leader sequence has been removed by means of translation, now we have the proinsulin having only segments A and B and also the C chain. You see, I removed the leader signal. What is called actually the leader sequence or the sequence what is known as the signal protein. Now once it is being removed, that is what is called the C chain has been removed, it is being excised by a process, and after that we have received the insulin hormone. The two chains of alpha and beta are A and B. The two chains are being linked together by means of disulfide bridge. So this is the insulin and this is the proinsulin and this is pre-proinsulin. So comparison, pre-proinsulin is formed of, that is A chain, B chain, C chain plus leader sequence. Then after the removal of leader sequence it is called proinsulin formed of A, B and C chains. Then after the C chain has been removed, we have formed the original insulin hormone that is formed of A and B chains only. This is what is called the nutshell, or what we have the brief note about what we have the extraction and processing of insulin hormone. Insulin is the first ever pharmaceutical recombinant drug that is normally administered to humans. Now the use of what is called actually this recombinant insulin, that is to be given to the patient with the diabetics, was started in 1982. That means the approval to the use of recombinant insulin, that is for humans, was given in the year 1982. But it was marketed under the trade name, that's Humulin. Humulin is a trade name for that hormone made through Recombinant Dna Technology only in 1986. So it was marketed under the trade name Humulin only in 1986. Now who were the first persons who extracted this insulin hormone? 2 persons in 1921, Best and Bandy, isolated insulin from the islets of Langerhans cells of dog and demonstrated their effectiveness against what is called the diabetes mellitus disease. So these were the 2 people just actually demonstrated the effect of what is called insulin hormone against what we have diabetes, and they were the first they extracted or isolated from the islets of cells of dog. Okay, that's about what we have this one. I'll show the recombinant technology how that is recombinant DNA for insulin hormone is produced. Now human insulin production using recombinant DNA technology. Now we have the genes for the insulin production present in the beta cells present in the beta cells of pancreas. Islets of Langerhans. Now this is the human pancreatic cell having the gene for insulin production. So human insulin producing gene. Now we have the bacterium, the normal bacterium used as a host for insulin production is E. coli bacterium. That is the best one, using E. coli bacterium. Now this is the E. coli bacterium. It has a plasmid as well as its own DNA. The plasmid is acting as a vector. So we have a number of vectors for the transferring the gene from one cell to another cell. So the most suitable vector is what is called the plasmid DNA, the circular double-stranded DNA present in the case of bacteria. Now, the plasmid DNA is cut open by using restriction endonuclease enzyme. So that what is happening, we are cutting this DNA. It's being cut open with the sticky ends. We have studied what is sticky end as well as blunt ends in biotechnology and botany. Now it is cut open. The same enzyme is also used to cut what is called the gene for human insulin production. So the same enzyme, what is called the restriction endonuclease enzyme, for cutting open the plasmid as well as for just removing the gene of interest, here the human insulin gene. For example, ECOR1, that is an enzyme used for cutting open. Now it has what we have the sticky ends. Now the next one, what we have to make the recombinant DNA by inserting what is called this gene of interest with the plasmid. Okay, the cut open is done by restriction endonuclease enzyme because they are acting as molecular scissors. And the joining of this cut open plasmid with what is called the gene of interest, here the human insulin gene, is by means of enzyme ligase. The product is called hybrid DNA, or we can say recombinant DNA. Then the recombinant DNA needs a host cell for its multiplication. So it is being inserted or introduced what is called into the bacterial cell, what I mentioned about E. coli bacterium, that is a recombinant bacterium, after introduction of what is called the plasmid DNA with the known gene. That's called recombinant DNA. Then it's called recombinant bacterium. So introduction of this recombinant DNA into bacterial cell. So we need large molecular insulin production. For that we need a fermenter. I mentioned already that is what is called fermenter is a process. Fermentation is a process and fermenter is a vessel, a large vessel where you have normally large mass, a biomass can be produced. Here the biomass is nothing but an insulin hormone. So to produce large molecule of insulin, large number of molecules of insulin, we are using what is called fermenter tank. So in that fermenter tank the bacteria are allowed, so they multiply and produce what we call the insulin hormone. Then the insulin hormone is normally having, I mentioned already, it is the pro-insulin. Prepro Insulin having the chains, the leader sequence, etc. The next event only extraction and processing of human insulin, removal of leader sequence, removal of C chain to get what is called the double, that is polypeptide chain of insulin hormone, what I already represent the diagram. So first one we are getting what is called Prepro Insulin. From that one we are removing the leader sequence, we are getting what is called pro-insulin. From that one the C chain is removed, now we are getting that is insulin hormone only with the two chains and both are linked together with the help of what is called disulfide bonds. That is a product what we are obtaining. The extraction and processing human insulin, I already drew the diagram separately. This is the part. Okay, this is the mechanism, just what we have normally, just during the production of insulin hormone by our DNA technology. You have already studied how to cut open the plasmid, how to rejoin the plasmid with the known gene of interest, etc. in biobotany. So I don't go there. So simply the incorporation. The cutting open is by means of restriction endonuclease enzyme. It is called a molecular scissor, and the joining of a particular DNA of known interest with the plasmid DNA by using what is called another enzyme ligase. That is called the paste molecule. So that's about what we call the insulin production and its use, etc. You have to go for the next one.